Hello viewers. Today we will talk about ethylene, the only gaseous hydrocarbon that is known to act as a very important plant hormone. Now, ethylene has a formula of CH2 double bond CH2. Its effects on plants were known long before the discovery of oxygen, and it goes back to 1800s when city streets were lightened by lamp that burned illuminating coal gas. It was known that leaks from the gas mains caused defoliation of shade trees along the streets. In 1901, Dmitry Neljubov demonstrated that ethylene was the active component that caused this. He also noticed that if pea seedlings are exposed to the illuminating gas, the stems grew horizontally. When the components of the illuminating gas were separated and individually each were tested for these effects, it was found that only ethylene can cause these effects and none other components could show this. Ethylene was active at concentration as low as 0.06 ppm in air. From Neljubov's findings and other studies, people realized that ethylene exerts a major influence in the growth and development of plant, including growth of most tissues, fruit maturation, fruit and leaf abscission, and senescence. The biosynthesis of ethylene starts with methionine, which is the precursor. Now, methionine is converted to S-adenosyl methionine using ATP by the enzyme adomate synthetase. Now, this S-adenosyl methionine is then converted to ACC, that is aminocyclopropane carboxylic acid. The critical enzyme is ACC synthase, and this is the rate limiting step of ethylene biosynthesis. This enzyme is inhibited by molecules like AOA and AVG. Factors that induce the activity of this enzyme are fruit ripening, flower senescence, auxin, stresses like wounding, chilling, drought, as well as flooding. Now, this ACC is finally converted to ethylene by the enzyme ACC oxidase, and this step requires molecular oxygen. This enzyme is inhibited by cobalt ion, anaerobic condition, obviously because this step requires molecular oxygen, as well as temperature above 35 degrees centigrade. This enzyme is induced during fruit ripening. The ACC, which is the immediate precursor of ethylene, can be conjugated with malonyl-CoA to produce N-malonyl ACC, which is the conjugated form of this. Now, the important component of SCH3 group of methionine can be recycled by the Young cycle and regenerate the methionine. This Young cycle recycles the CH3S group of methionine so that it is conserved for continued synthesis. Now, plants produce ethylene in response to different types of stresses such as drought, flooding, mechanical pressure, injury, and infection. Ethylene is also produced during fruit ripening and programmed cell death and in response to high concentrations of externally applied auxin. Actually, many effects of auxin are now known to be due to auxin-induced ethylene production, such as inhibition of root elongation. Imagine a pea seedling pushing upwards through the soil and coming to a stone or a pebble as an obstacle. As the tip pushes against the obstacle, the resulting mechanical stress induces the seedling to produce ethylene. Now, this hormone then instigates a growth maneuver known as triple response that enables the shoot to avoid the obstacle and then grow normally. The triple response contains three components: slowing of stem elongation, thickening of the stem so that it becomes stronger, and a curvature that causes the stem to start growing horizontally. Now, as the effects of the initial ethylene pulse lessens, the stem resumes its vertical growth. If it again contacts a barrier, another burst of ethylene is released and the horizontal growth resumes. Finally, if the upward touch detects no solid object, then ethylene production decreases and the stem, now clear of the obstacle, resumes its normal upward growth. Now, it is ethylene that induces the stem to grow horizontally rather than the physical obstruction itself is shown by the fact that when ethylene is applied to a normal seedling in dark, growing free of any physical impediments, they still undergo the triple response. Ethylene can also induce rapid stem growth in some semi-aquatic plants, like deep water or floating varieties of rice. When they are submerged, particularly the young rice plants, during the monsoon, it triggers an increase in the ethylene synthesis. The resulting ethylene-induced internodal elongation provides a mechanism for the rice plants to keep pace with rising flood water. In tomato, if it is flooded, then it causes increase in the ACC, in the xylem sap, as well as increase in the ethylene in the petioles. Now, ACC is synthesized in the root, but it cannot produce ethylene or cannot be converted to ethylene, because if the root is submerged, then there is no supply of oxygen to convert ACC to ethylene. Consequently, ethylene will be transported via xylem sap to the shoot, where it will be converted to ethylene, and this ethylene, being a gas, cannot be transported, but it will diffuse in the local area, causing elongation of the stem. Ethylene may promote creation of air space in the root in the case of flooding. Normally, soil has air pockets through which plant can take up oxygen via root. But if the root is flooded, then it cannot take up oxygen, leading to a hypoxic condition. This hypoxia then induces activity of ACC synthase as well as production of ethylene in the root. This ethylene can induce cell death or cell separation and formation of aerenchyma, air channels, through which oxygen can move into the roots. Consider a leaf shedding in autumn, or death of an annual after flowering. Or consider the final step in the differentiation of a vessel element, where all the living contents are destroyed, leaving behind a hollow tube. These events involves senescence, that is the programmed death of certain cells. Cells are genetically programmed to die on a schedule. At the molecular level, the onset of programmed cell death, which is called apoptosis, is associated with a very high activity within cell that involves expression of many new genes. During apoptosis, newly formed enzymes break many chemical components such as chlorophyll, DNA, RNA, protein and membrane lipids. Plant can salvage many of the breakdown products. A burst of ethylene is almost always associated with the apoptosis of cells during senescence. Senescence. Loss of leaves from deciduous trees is a very important process because it prevents desiccation during seasonal periods of climatic stress that severely limit the availability of water to the roots. Now, before the dying leaves abscise, many essential elements are salvaged from them and stored in the stem parenchyma cells. The nutrients are recycled back to the developing leaves the following spring. Autumn leaf color is due to a newly made red pigment as well as yellow and orange carotenoids that were already present, but now they are visible because the dark green chlorophyll is broken down. When an autumn leaf falls, at the breaking point, there is an abscission layer that develops near the base of the petiole. The small parenchyma cells of this layer have very thin walls which facilitate this abscission, and there is no fiber cells around the vascular tissues. The abscission layer is further weakened by expression of enzymes that hydrolyze the polysaccharides in the cell walls. Finally, the weight of the leaf as well as the flow of the wind causes the separation within the abscission layer. Now, before the leaf falls, a layer of cork forms a protective scar on the twig side of the abscission layer that prevents infection of pathogens. Here you can see that there are 2 to 3 layers of cells in the abscission zone where there is expression of enzymes that breaks down the cell wall polysaccharides. So these cells, they become protoplasts, they round up, increase in volume and thereby push apart the xylem tracheary cells, facilitating the separation of the leaf from the stem. Now all these events are highly induced by ethylene. In this experiment, 2 plants are shown. On the left, there is the wild type birch, and on the right, there is a mutant birch where the ethylene receptor is mutated. So, this plant becomes ethylene insensitive. Now, both were exposed to ethylene, where in the wild type we can see almost complete defoliation, but in the mutant, which is ethylene insensitive, there is no leaf abscission. The main factor that controls leaf abscission is a change in the ratio of ethylene to auxin. Here you can see that in the leaf maintenance phase, the auxin level is high and that reduces the sensitivity of the abscission zone cells towards ethylene. So, there is no leaf shedding. Now, during the shedding induction phase, the level of auxin goes down. Consequently, the abscission zone cells become more sensitive to ethylene as well as there is more production of ethylene. This triggers the shedding phase, that is, this triggers all the different events associated with abscission that we have discussed so far. And finally, in the shedding phase, the enzymes that break down the polysaccharides of the cell walls, they are highly expressed and consequently there is cell separation followed by leaf abscission. Immature fruits, they are usually tart, hard and green. These features help protect the developing seeds from herbivores. But after ripening, the mature fruits, they help attract animals that disperse the seeds. In many cases, a burst of ethylene production in the fruit triggers the ripening process. Enzymatic breakdown of cell wall components within the fruit softens the fruit. On the other hand, conversion of starches and acids to sugars makes the fruit sweet. At the same time, production of new scents and colors helps advertise ripeness to animals which are attracted, eat the fruits, and disperse the seeds. This graph shows the different changes during fruit ripening. As we can see that cell division and cell expansion ceases. Eventually, there is accumulation of ethylene, increase in respiration, that will come back soon to this, and increase in the amount of pigments, which is perfectly correlated with the process of fruit ripening. This graph shows changes in the ACC oxidase activity, amount of ACC, as well as amount of ethylene during ripening of golden delicious apples after harvesting. Here we can see the increase in all these components, they perfectly correlate with the timing of ripening after the days of harvesting. During ripening, a chain reaction occurs. Ethylene triggers ripening. At the same time, ripening triggers more ethylene production, resulting a huge burst in synthesis of ethylene. Now, since ethylene is a gas, the signal to ripen can spread from fruit to fruit. If we take a green fruit and enclose it in a paper bag, we will be able to speed up the ripening process, because then the ethylene will accumulate in the enclosed environment. There are many fruits such as tomatoes, avocados, bananas, peaches, apples, pears. In these cases, during ripening, there is a rapid and large increase in cellular respiration, as shown by huge increased uptake of oxygen and a high production of carbon dioxide. This phase is known as climacteric, and these fruits are called climacteric fruits. On the other hand, fruits like citrus fruits or grapes or cherries and strawberries, they show a steady or gradual ripening, and they are called non-climacteric fruits. In climacteric fruits, increased ethylene synthesis always precedes the increased carbon dioxide production, and this increased ethylene synthesis is responsible for accelerating all the processes that are associated with the ripening and that we have discussed so far. Ethylene acts via receptors. These ethylene receptors, they can be divided in two subfamilies, subfamilies 1 and subfamily 2. They are very similar to the bacterial two-component response regulators. There is an ethylene binding domain. There is a GAF domain, which is conserved cyclic GMP binding domain. There is a histidine kinase domain, and there is a receiver domain. Based on several experiments, model for ethylene signaling in Arabidopsis have been proposed. The ethylene receptor seen here, which is ETR1, there are many more, acts as a dimer, and it is inserted into the membrane of endoplasmic reticulum. As you can see, the dimer is held by disulfide bond, and it contains a copper cofactor. The binding of the copper cofactor is mediated by the RAN1 protein. Now, ethylene binds to the receptor via this copper cofactor. Now, whenever there is no ethylene, the receptor is active. This active receptor uses its kinase activity and activates a downstream effector called CTR1. This CTR1 is a kinase that via the MAP kinase pathway can inactivate another membrane-bound factor called EIN2. Consequently, the downstream effects are negatively regulated and there is no ethylene response. But when ethylene binds to the receptor, then the receptor is inactivated. Consequently, CTR1 is not activated. Consequently, EIN2 can become active. This in turn then activates transcription factors within the nucleus such as EIN3 and ERF1. These transcription factors then trigger the ethylene response genes and we see the different effects of ethylene that we have discussed so far. Here we see an ETR1 mutant in Arabidopsis. Since the receptor is defective to bind ethylene, we can see the mutant is completely insensitive to ethylene and doesn't show the triple response. This is a CTR1 mutant. Here, the downstream effector of the receptor is inactivated. Consequently, the ethylene response genes are always on and this becomes a constitutive display of the triple response. There are different bioassay methods of ethylene. All are based on its physiological effects. For example, the triple P test is based on the physiological effect of triple response on etiolated pea seedlings. The pea stem swelling test is more quantitative, where the pea seedlings are used to measure the ethylene concentration by a marked increase of stem swelling expressed as a ratio of weight to length. Even in 1 ppm of ethylene, the ratio is about 4. There are so many applications of ethylene. It's one of the most widely used plant hormone in commercial purpose. There are many kinds of fruits that are ripened in huge storage containers in which ethylene levels are enhanced. On the other hand, it is also used to slow ripening caused by natural ethylene. For example, some fruits such as apples can be stored in bins rich in carbon dioxide and with constant air circulation. The constant air circulation prevents accumulation of ethylene and the carbon dioxide inhibits the synthesis of new ethylene. Fruits stored in this way can be picked in autumn and can still be shipped to grocery the following summer. Ethylene is also used commercially to promote fruit loosening in cherries, blackberries, grapes and blueberries, thus making mechanical harvesting possible. Ethylene is also commonly used as a fruit thinning agent in commercial orchards such as in prunes and peaches. Now, ethylene is one of the widely used plant hormone in agriculture, but there is a limitation because being a gas and having a very high diffusible rate, it has to be used in an enclosed environment. It is very difficult to be applied in gaseous forms in the field. So, this problem has been overcome because nowadays there are synthetic chemical compounds are available which can be sprayed on plants in aqueous solution. The plants will then absorb and transport these chemicals and then within the plant they can be broken down to release ethylene. One such common compound is ethyphon, 2-chloroethyl phosphonic acid. The trade name is ethyl. Ethyphon is stable at low pH, but at pH 4 and above, it is slowly broken down to release ethylene. Since the pH within the plant is above 6, ethyphone is easily converted to ethylene and can exert the hormonal effect. The conversion of ethyphone to ethylene is non-enzymatic. It is a simple base catalyzed reactions releasing ethylene and phosphoric acid and chloride ion as byproducts. The breakdown of ethyphone into ethylene is a very slow process and may continue for several days in the plant cells. Now, given the importance of ethylene in the post-harvest physiology of fruits, the genetic engineering of ethylene signal transduction pathways have potential commercial applications. For example, by engineering a way to block the transcription of one of the genes required for ethylene synthesis, molecular biologists have created tomato fruits that ripen on demand. These fruits are picked while green and will not ripen until and unless ethylene gas is applied. As these methods will be refined, they will reduce spoilage of fruits and vegetables, a problem that ruins almost half of the produce all over the world. So viewers, we have talked about ethylene, the only gaseous hydrocarbon that can act as a major plant hormone. We have seen its effects in many different aspects of plant growth and development, making it a very important plant hormone, and we have also seen that it is commercially extremely important. Thank you.